Hey guys, welcome. My name is Tanner Smith. I'm a freshman at the University of Alabama. I'm majoring in biology. So I'm gonna make these little lecture videos from here on out for the rest of the semester, just to kind of help me recap, help a couple friends study for the test, and anyone else who gets benefit from it, congratulations, enjoy. I'm going to start off with this first chapter a little bit into the introduction to biology, but with cellular respiration. Cellular respiration is really, really important for all cells, prokaryote, eukaryote, plant cell, animal cell. Everything uses cellular respiration. And what is the point of it? It's to make energy, to create ATP. Now there's only two ways to make ATP. Total, period, that's it. Two ways. And that is oxidative phosphorylation and substrate level phosphorylation. Your oxidative phosphorylation is going to be very complicated, and substrate level phosphorylation is going to be a little more simple. We'll go into a lot more detail on that later, mainly as I go into more detail on it. I'll go into more detail on it on here. But for now, we're just going to know just one's more complex and one's more simple. And oxidative, which is the more complicated one, is also going to have a higher yield. It's going to produce more ATP. Okay, so now we're going to go in. Quick summary, some major topics. You can read over those. We're only going to cover very, very surface right now, just up to pyruvate oxidation and barely into that. We're mainly going to stop it. We're going to go into detail on glycolysis. We're going to touch a little on pyruvate oxidation. So going down, all life needs energy. We eat for energy. So autotropic organisms, so that's plants, convert sunlight into energy. Heterotrophs obtain energy catabolically, so by eating. So we're obviously, which one? Heterotrophs. Sun is the ultimate source of energy, though. That's where all of it comes from. It's just recycled around. So anabolic organisms versus catabolic organisms. Anabolic process of photosynthesis, catabolic eat food via the breakdown of organic nutrients. That's how we get our energy. They are inverse processes of each other, photosynthesis and cellular respiration. So if you look at the chemical formula, what do you have? You have sugar, C6H12O6, and oxygen in the air, and that yields carbon dioxide, water, ATP, and heat. What's photosynthesis? What's the back of that? Sunlight energy, which is your energy and heat, and you have carbon dioxide and water, and you're going to produce sugar and oxygen. So if you know photosynthesis from one class, or you know cellular respiration from another class, just know they're basically backwards of each other. They're complementary. Glucose plays an essential role in cell metabolism. The cell must use the energy stored in food molecules to make ATP. You can read over these cells. I'm just going to try to cover the important stuff, so if you see something interesting, feel free to pause the video and read over it. Two very important pathways. Here we have oxidation and reduction. So oxidation is the removal of electrons. Reduction is the addition of electrons. That's typically done by adding and removing hydrogen atoms. So, hydrogen atoms have one electron, makes it really easy. Here we go in. We have simple redox reaction here. Just see it's going to charge the two. They're both going to come out of it with a charge. It's a very positive marker, no pun intended, of a redox reaction. Going into cellular respiration in terms of a redox reaction. So glucose is oxidized because it's losing an electron, and then oxygen is reduced because it's going to be gaining an electron. Both of those are in the form of hydrogen. Like I was telling you earlier, it's most common, especially in organic, and you're really only working with 6 or 7 key elements. So hydrogen is going to be the main one for adding and reducing charges. Excuse me. I broke it. Okay. All right, so here we go. We're going into the first, there's 3 steps of cellular respiration that you need to know. Excuse me while I try to get my computer to obey me. But for now, we have to know there's glycolysis, there's the citric acid cycle, and there's the electron transport chain. And there's another often neglected step in there. That is the oxidation of pyruvate, okay? And the reason it's not usually listed is because it's kind of an assumed step. It's going to be in there, but it doesn't even take place in anaerobic. It only takes place in aerobic organisms. So hence the aerobic meaning air, so anaerobic without air, oxygen. So ATP is all we're doing. That's what we're trying to create, is ATP. All right. But we're going to go into all of the products and all of the reactants, everything that goes in, everything that comes out of cellular respiration. Again, starting with glycolysis because that's the first step. So what do we have in glycolysis? What's the first step? Cellular respiration. So what do you have on your reactant side of cellular respiration? Just glucose, just the sugar. You got C6H12O6. So you got a glucose coming in, and you're going to have to try to make it into some other stuff. Well, you got to start by taking some energy in. Where are we getting energy? Or we're trying to make energy. Well, we have to borrow energy, and anything that's not listed in the formula, so anything that we're not given that's actually going to be taken up by cellular respiration, is going to come from the cytosol. And at the end, we're going to have to return everything back to cytosol that we took from it. It's like a bank. We can borrow from it as much as we want, but we have to return whatever we take out of it. So glycolysis, the very first step in converting from glucose to glucose 6-phosphate. Okay, I'll explain what that means in a second. You have glucose, which is just a 6-carbon sugar, basic sugar. You have ATP from the cytosol, and you have hexokinase, which is the first enzyme we're going to cover. Now, enzymes have a handful of root words you need to know. So kinase, any enzyme that contains the word kinase means it's got the ability to add or remove phosphates. So if you think about it, hexokinase is going to have the ability to add or remove a phosphate to the 6-hexocarbon. Makes sense because we have glucose and this ATP coming in. That hexokinase is going to split the ATP into ADP and a phosphate. So the ADP is going to be released out into the cell, and that phosphate is going to be attached to the glucose cell we have onto the 6th carbon. Hence the name glucose 6-phosphate. Okay, so now going into the next step, step 2, we use another, this is called an isomerase. Don't worry about the name of it, it's phosphoglucoisomerase, which makes sense because it's phosphate and glucose, but it's not a very commonly used enzyme. You shouldn't have to know it. If you do, though, like I said, it's very self-explanatory, phosphoglucoisomerase. Again, the root word for this enzyme is isomerase. And isomerase, sounds just like its name, it's used to change between isomers, all right? So you're going from, in this example, from glucose to fructose. Everything else stays the same. You keep a phosphate on the 6th, so on the 6th carbon. So you go from glucose 6-phosphate to fructose 6-phosphate. You're going to go on, that's through step 2. Now step 3, you have phosphofructinase, fructokinase. And what did I tell you about kinases? All kinases means they have the ability to add and remove phosphates. Okay? Well, again, if we're going to have phosphate, we're probably going to have it in the form of ATP, especially in all organic, all living organisms. So your ATP, again coming from the cytosol, is coming in. And we have a fructose 6-phosphate. Each of these are different reactions. So the reactants in this one is fructose 6-phosphate, ATP, and your enzyme used in the reaction is phosphofructokinase. And we're going to end up with fructo-1,6-biphosphate. If you're picking up on the trend here, I don't know if you know what that means, but you have your fructose, still your base, which is an isomer of glucose. You're still moving on down the chain from one to the next. And you're going to have a phosphate on the 1st and the 6th carbon, which makes sense. Fructose 1,6-biphosphate. Biphosphate just means 2 phosphates now. Now the next step is where things start to get a little more fun. We have aldolase, okay, aldolase if you'd like, and that actually splits this fructose 1,6-biphosphate into 2 totally separate products. Into what we're just going to know them as GAP and DAP because they're too big for me to try to pronounce in this introduction to biology. So we're just going to have GAP and DAP. Out of these 2, you need to know that GAP is unstable. It's inherently unstable. Okay? And GAP is going to be, with a G, GAP is going to be higher energy. So this isomerase at the end, which is again dealing with isomers. These are isomers of each other. So we're dealing with isomers. So step 5 is an isomerase converting DAP to GAP. Real simple. So now going into step 6, we're just going to have 2 gaps. That's the whole thing. Everything for all of step 1 through 5, which is broken down into part 1 for this slide's purposes, we're converting glucose, we're taking in 2 ATPs, and we're producing two gaps. Remember, you have two separate gaps now. Now, if you look at gap is two separate 3-carbon molecules, which makes sense because we started with a 6-carbon molecule, sugar. So if you pay attention to the chemistry of it, it all works out. I don't know if you need to or not. It's up to your specific class. Going into part 2, we're just going to start with step 6. We just have 2 gaps. So each of these gaps are going to perform the exact same way throughout the rest of the reactions, which means all of our products for every step is going to be doubled. We're going to have 2 NADH, we're going to have 2 ATP, we're going to have 2 H2O, 2 ATP, and 2 pyruvates. Everything's doubled from here on out. So, step 6, we're converting each of our 2 gaps with a super fancy enzyme, which, again, you don't need to know this in much detail, but it's a dehydrogenase. Which, what does that mean? Dehydrogenase, D without hydrogen, or hydrogen. Broke that down a little too much. So we're just taking hydrogen off of our gaps. Where's that hydrogen going to go? We have NAD+, again in the cytosol, because everything that's not in our formula comes from the cytosol. So 2 NAD+ coming from the cytosol, binding with those 2 hydrogens that come out of this dehydrogenase in step 6 to produce 2 NADH. All right, you're going to use those later. You'll see where they come in. So you have 2 NADH released as our very first overall product. That's going to stay released. We're not going to use that for anything else in the rest of glycolysis anyway. And that's going to produce not only 2 NADH, but it's also going to produce a 1,3 biphosphate glycerate. Biphosphoglycerate. Again, sorry I don't know these words too well. I just learned this stuff today. But I don't think you're going to know this much vocabulary of it, just the basic breakdown of what's being produced. I'm just overly complicating this. So we're going to go down. Next step, we're going to have another kinase adding, and that's going to be dealing with phosphates. This time, though, we're going to have 2-ADP from the cytosol. We already have those phosphates. Remember, it says biphospho, means 2-phosphate, and they're on 1 and 3. They're on the first and third carbon. So we're going to bond with these, and we have 2-ADP coming in from the cytosol, with this kinase pulling our phosphates off of our biphosphoglycerate and attaching to make ATP. It's another one of our overall products. It's not going to be used in the rest of glycolysis. Now, if you remember in the step in part 1, we took in ATPs from the cytosol. So now we've produced 2. Remember gaps, we've doubled everything we did. So we're producing 2 ATP right here. We can return our debt to the cytosol. Now, everything we borrowed from the cytosol has been returned. Everything else we get from here is just net. It's all net products. Okay. So going in, step 7 is just going to be a debt settling step. So when the ATP comes out, which is what I just said. Then we have 8 and 9. So the only thing you need to know out of 8 and 9, I'm not going to go into the molecules, not going to go into the chemistry. Really not too important for intro to chem. They're going to nail you with it in cellular respiration or in cell bio. But the important thing you need to know, out of 8 and 9, you produce H2O. Everything comes in pairs because you had 2 gaps coming in. So you have 2 H2O, 2 water molecules getting released. And then going into step 10, you have another kinase, and this is pyruvate kinase, which is a convenient name because you produce pyruvate, and the kinase, dealing with phosphates, 2 ADP from the cytosol come in, produce 2 ATP. So you have 2 ATP and 2 pyruvate. Now, each of those pyruvates are 3-carbon molecules, which makes sense again because we started with a 6-carbon sugar. Real simple. So what are your overall products? We have 2 NADH, 4 ATP, and 2 H2O, and 2 pyruvate, which is also known as pyruvic acid. Remember, we don't actually have 4 net ATP. We've produced 4 total, but net, we've only produced 2. Because if you remember in part 1, we had to borrow 2 from the cytosol to get this whole chain started, to get it going. So now you have these 2 pyruvate. Well, what do you do from there? Well, pyruvates have to be oxidized in order to be used in the citric acid cycle. Well, anaerobic, they cannot oxidize it. That's their thing, anaerobic, without air, without oxygen. So they are unable to oxidize the pyruvate. So what do they do instead? They do a process called fermentation. And that's actually going to be the start of next week's lecture. We're going to go into fermentation and it's going to use NAD+, can be all fancy. And we're going to go into how aerobic organisms, which with air, oxidize this pyruvate and bring it in for the cellular or for the citric acid cycle. Again, that's all going to be next week, so I'll put that on part 2 of the video and hopefully have that up by this Thursday. All right. Thanks for watching.